Hi there, I'm Chris Mancher, and I'm a master's student in the Department of Biological Sciences at the University of Manitoba. I come from a family farm in a small town called Giver Plains and have a degree in agriculture where I studied plant biotechnology and crop protection. Across Canada, most farmers utilize fungicides to protect their crops from fungal pathogens to secure a steady food supply for our growing population. However, fungicides aren't always a magic bullet that can solve these problems. Some pathogens are becoming resistant to these chemicals, and there is evidence of some fungicides leaching into the surrounding environment where they could affect off-target organisms. If fungicides are no longer a viable option or become banned, we'll need to find an effective alternative. So, let's do some brainstorming. We need to develop something that 1. Degrades readily in water and soil, 2. Affects only our targeted pathogen, and 3. Is compatible with our current application systems like spraying. I think I know the solution.
Okay, now here's the plan. Currently, fungicides work by inhibiting proteins or enzymes in the fungus, but instead, let's stop the RNA from making them in the first place. In almost every organism are enzymes that chop RNA depending on the template provided. So let's create a template that targets RNA responsible for important processes, like cell wall synthesis or proteins related to infection. All we need to do is introduce that sequence to the fungus, and then that gene is now silenced, and fungal growth is reduced.
This concept is called RNA interference, or RNAi for short. This species-specific control method based on RNA sequences has been shown to reduce infection in many species, and some pathogens like Sclerotinia stem rot and Botrytis gray mold has been controlled right here in this lab. This brings me to my own research I've been doing over the past year. Downy mildew is an algae-like fungus that affects many crop species, from cucumbers to soybeans and even grapes. Control options are limited, which makes this disease a perfect candidate for RNAi. What I've been working on is developing a bunch of different RNA sequences that I believe could silence important genes, and then testing those formulations on small seedlings.
First, I spray the treatment on the leaves, inoculate it with fungal spores, and then place it in a cool and humid growth chamber. After a week, I collect the leaves, image them with a stain to take a closer look at the infection, and then extract genetic data to determine if those genes were silenced.
While my experiments are still underway, RNAi is showing a lot of promise against different pathogens and could be a great way to provide fungal control in a safe and environmentally friendly way.